So today for the Journal Club, we were going to focus down on several papers in the mid 60s, and in particular, two papers from 1965 that initiate the whole field of area of lymphokines, which subsequently was broadened to become called cytokines, because there was a realization down the line or a few years later that other cells besides lymphocytes could make very similar molecules. So it was broadened into cytokines. But lymphokines is very, very important for immunology because they were the key, as hopefully I will go on and demonstrate for you, the key to how the whole system is regulated. The question was, how does the system know that it should recognize and respond to the invasion by some foreign thing, microbe or whatever? And should it respond a lot or a little bit? And when is it over? And how does it begin? All these different kinds of questions were impossible to approach unless one invoked lymphokines. So the first paper we're going to discuss is by Shinpei Kasakura and Louis Lowenstein from the hematology division in a Department of Medicine at McGill University in Montreal. The title of the paper is "A Factor Stimulating DNA Synthesis Derived from the Medium of Leukocyte Cultures." You notice that they used the word leukocyte rather than lymphocyte because they weren't sure what kind of cell was actually making these factors that they had talked about. It appeared in Nature in 1965. And just to give you a little insight into these two fellows, Lowenstein was the leader of the lab. He was a hematologist at McGill, whereas Kasakura was a postdoc who had come from, he was also a hematology type, and he'd come from Japan for training. So he was the younger guy. Lowenstein was the older guy. And I've actually met, I never met Lowenstein. He's maybe 10 or 15, 20 years older than me, but I met Kasakura and visited him in Japan. And he took me to the Japanese opera, No opera, which was memorable. And we went out to dinner and we had all kinds of things. And one of the things in my book is a picture of myself together with my son, Kerry, and Kasakura on one of the visits that we did to Japan. So in the introduction of this paper, and of course, they mentioned the first thing they talk about a little bit about the history of why they did the experiments that they did. So they start with Peter Noel's paper in 1960 that demonstrated that monocytes, lymphocytes, large mononuclear cells could undergo DNA replication and mitosis, because up until that time, it wasn't really established that they could do this. Once that was discovered and published in 1960, so it's 5 years later now, other people in between 1960 and 1965, there were several different groups involved in this whole thing. One was a group at NYU that was led by a cytogeneticist by the name of Kurt Hirschhorn, who had latched on immediately to the whole deal that Peter Noel had presented, because he was a cytogeneticist. And this idea that you could get normal cells to undergo DNA replication and mitosis and was really important for the cytogeneticist, because then they could look at the chromosomes under the microscope, which is what they did for a living. So Hirschhorn wondered and had a paper out that came out in 1963, before these two that I'm going to talk about today, where he asked the question, well, if PHA, phytohemagglutinin, can make these cells proliferate, what about specific antigens? Is this an immunologic reaction? That was his question. And so he tested several different antigens, purified protein derivative of tuberculosis, diphtheria, toxoid, pertussis, and so forth. And he published a paper in 1963 in Science that said, yes, the cells will proliferate in response to these specific antigens, only if the person had been previously immunized or infected with that particular organism. That was very important, because that's immunologic memory. That's one of the major characteristics of immunity. And that that together with another report from a hematologist by the name of Barbara Bain from 1964. And all these papers came out in either in Nature and Science, and they're all 1 or 2 pages. So if you really want to get into this stuff, you should read these papers. Although, as I'll get into, they're not as easy as you might think. Barbara Bain, who was interested in transplants and the whole transplant immunology end of things, and she mixed together 2 lymphocytes from 2 different people, and found that would then activate the proliferation of lymphocytes. So these 2 different phenomena that was tacked onto the PHA phenomenon really blew the lid off. Everybody got excited, particularly the transplant immunologists. Because they wanted to know if you could predict beforehand whether a skin graft or a kidney graft placed onto a person was going to be rejected or wasn't going to be rejected. What they knew at the time was that if you tried to, you could exchange skin grafts between identical twins, but everything else you did between 2 different people would be rejected. And so they had published, Barbara Bain and her, and Lowenstein actually had published this before this paper with Kasakura and Lowenstein came out in 1965 in Nature. That's the background. And one of the things that Lowenstein and Kasakura mentioned was that it had been reported within the past year or 2 that histocompatibility antigens could be released into the culture medium if you cultured lymphocytes both in dogs and from rabbits. Then if you looked in the culture medium, you could find soluble antigens in the histocompatibility antigens, HLA antigens, as we call them. And so they wondered whether or not you could take supernatants from these mixed cultures, mixed lymphocyte cultures, MLCs, and whether you could take, there might be antigens in the supernatant that would stimulate the cells. You wouldn't have to mix 2 different cells together. You could just take the soup from these reactions and so forth. So they went looking. That's why they did the experiments that they did. And so in the methods of this paper, they took peripheral blood leukocytes, white blood cells, not trying to select out lymphocytes or monocytes or polymorphonuclear leukocytes or whatever, and they put them into tissue culture 199 that we've talked about before with penicillin and streptomycin, so we didn't get bacterial overgrowth. And they cultured them for 1 day, for 3 days, for 5 days, and then they would spin out the cells with a low-speed centrifugation at around 10 or 15 minutes, remove the supernatant and remove the cells, and then that would be their medium. That would be one thing that they were going to test. And then they took an aliquot of the supernatant and spun it in an ultracentrifuge at 100,000 times gravity for an hour, precipitating whatever was big stuff in the medium. And then they took the medium off of that and they called that soup. So they had medium and then they had soup, and then the precipitate from the ultracentrifugation in the bottom of the tube, they resuspended into the original volume that had been spun down. And they labeled that P, that was the precipitate. And those are the 3 different things that they tested to see whether or not they had mitogenic activity, mitogenic meaning that it would cause the cells to proliferate. And they would, in order to test that, they would put them onto either autologous lymphocytes or mononuclear cells that they took from individuals. That means all the soups and all the things that they were testing was tested on to the same person's cells, or they could test them on somebody else's cells. And that would make them allogeneic. And when you mix allogeneic cells together from 2 different individuals is when you got this mixed lymphocyte culture reaction and proliferation and so forth. And so what they found was when they did this, was that when they tested the medium, the supernatant from the ultracentrifuge and from the precipitate, that they also sort of contained mitogenic activity when they tested them on allogeneic lymphocytes, causing those cells to undergo a proliferative response. But when they tested cells from, not from mixed cultures, but from just single cultures, they, and as long as they tested them on autologous cells, they were negative. They did have some activity on the allogeneic cells, and they didn't know really what that was all about. But they always had more activity when they had the mixed culture, supernatant and so forth and so on from the mixed cultures. So so that was it. And they have just a couple of tables in this paper, and the way they wrote the paper and the data in the tables is really difficult to figure out exactly what these guys found and how they interpreted it. I mean, I've been reading these kinds of papers for a long time, let me tell you. And this was not easy. It was only 2 pages. You know, you'd think, I thought, oh, it's a piece of cake. And I already knew what the conclusion was. Anyway, in the discussion of this paper, they said that the cultured leukocytes released a quote unquote blastogenic agent into the medium when tested on allogeneic cells. But they had no data on whether the so-called factor that appeared in the supernatant of these cells was actually produced by the cells, or whether it was released by dead and dying cells within the cultures, which could possibly happen too, because over these several day period, you know, some cells would drop off and then they disintegrate, and you'd have the remains in the culture. But after all was said and done, and after they'd done these experiments, they said, well, maybe whatever it is are really transplantation antigens, which of course is what they were looking for. They wanted to find what they were looking for. And you got to be careful in science, because most of the times, if you look hard enough, you can find what you're looking for. Now, whether it's true or not is another question, you know? And so, but the other interesting thing about these 2 papers that we're talking about today is that the second paper that appeared back to back, right together in Nature, was by 2 people by the name of Julius Gordon and Lloyd MacLean, also from McGill University. So what's that all about? They couldn't all get together and publish a paper with 4 authors? Why are there 2 separate papers from McGill? And I asked that to Kasakura several times, and he wouldn't tell me. You know, he gave me this inscrutable Japanese, you know, like, I don't know. And I know that there's a story about that. And I think you can discern that from reading both of these papers together, which I'll tell you about right now. From the standpoint of the background of this paper was MacLean, Lloyd MacLean. He was the mentor. He was the chief of surgery. And this kind came from the transplant unit, not hematology. Barbara Bain and Lowenstein and Kasakura all came from hematology. So these guys came from surgery. And usually the medicine people and the surgery people don't talk to one another. Or if they do, they don't like each other. And so that's probably what was going on here, but I can't tell one way or the other. In their introduction, they wondered whether or not there is a mitogenic factor really released by these cells when you did a mixed lymphocyte culture. They were interested in the mixed lymphocyte culture as a possible predictor of the disparity of the histocompatible nature of tissues from one individual and another one. And they wanted to hopefully use it to be able to type whether or not you could do a kidney transplant or a skin transplant or whatever. So in their materials and methods, they cultured both single lymphocytes or mononuclear cells from a single individual, as well as doing a mixed lymphocyte culture, just like Kasakura and Lowenstein. And then they made several different test samples from these cells and the supernatants from these cultures, these either single cultures or mixed double cultures. First of all, they'd take the culture fluid, they would centrifuge it, low speed centrifugation to get the cells out, take the soup off, and that was the supernatant. Then they wanted to know whether or not if you just killed the cells, whether they would release what they thought were the histocompatibility antigens into the soup. So they freeze thawed them. They also sonicated the cells with ultrasound, and they also subjected the cells to distilled water lysis so that they would pop become fat Alberts and then pop because they would take on water because they had salt on the inside. And finally, they heated the cells at 50 degrees for 5 minutes. And so all of those things they then tested, just like Kasakura and Lowenstein had done on single cultures. And so they found activity from their single cultures that were active, so of the whole thing, but the mixed lymphocyte culture, medium and so forth, was markedly positive. That was number one. And when they tested the whole thing on autologous cells from the same donor, there was no reaction. It was just the same thing that Kasakura and Lowenstein had shown, which is what they wanted to find, because they wanted it to be antigen specific for the disparity between the one individual and another individual. And the final thing is that the medium was positive, but all of the things that disrupted the cells, they freeze thaw and the heating and the lysis and sonication, that destroyed whatever it was. They also did experiments with some metabolic inhibitors of protein synthesis on the one hand and of DNA replication on the other hand, and both of those things blocked the appearance of any of the activity going into these things. So in the discussion of these papers, first of all, they called it a factor. That's where that's where this factor business comes in. Factor is a very amorphous term. If you look it up in the dictionary, it means we don't know what it is. So the factor is released by leukocytes in the cultures. So they're not saying it's a lymphocyte or a monocyte or whatever kind of a cell it is. They had done testing several days worth of this thing, and they found that the mitogenic activity was released over at least 3 or 4 days daily. They could replace the medium, and because of the fact that they couldn't find it in any of the lysed cells, or because of the fact that both the protein synthesis inhibitor and the DNA inhibitor prevented its release, they said it had to have been produced by the cells, needing metabolic energy and so forth and so on, and it wasn't just already preformed like a histocompatibility antigen would be, and then released when the cells were lysed. So that was it. And those 2 papers in 1965 set off cellular immunology, really, that then took over the next 15 years, from 1965 to 1980, and led to all kinds of discoveries because people could finally fiddle around with the whole immune system in the test tube or in the culture flask, and take some cells out, put some cells in, and do all kinds of things. They also set the stage for, so what are these lymphokines? What's this lymphokine thing, these factors? And I've said in some of my previous videos that it was a bipolar reaction in the immunologic community. They were the old school immunologists that were really into immunochemistry and the molecular nature of antibodies. And as far as they were concerned, nothing else made any or important at all. And then there were these new molecules or factors or whatever they were, needed to be characterized and needed to be figured out what they were. And for the most part in the United States, they basically discounted the lymphokines. They said, whatever. Weren't very many investigators initially interested in these sorts of things. And most of the new people came from different walks of life, like the transplant people, the surgeons and the hematologists and the cytogeneticists and all these other different kinds of people. The card carrying immunologists and whatever, a bunch of phenomenology. And so that really set the stage. I think that as time goes on, we'll see how everything evolved. So if you like this video, don't forget to like it on the YouTube channel, subscribe to the YouTube channel. And if you want to get notices from the YouTube people that Smith has put a new video up, you got to ring the little bell that's right next to the subscription thing. And I didn't know that initially, but I figured that out myself. And so you got to ring the little bell, and then you'll get a notification every time. And I'm making them once a week. So every every Monday morning. You can wake up and say, I wonder what Kendall's got to say today. So there's that. Then on the website, if you go on the website and go on the homepage and scroll down, you'll find the newsletter site. So sign up for my newsletter where I'm serializing my new book. It will keep the conversation going. So thank you very much. It's been great.